In order to understand the clinical use and monitoring of neuromuscular blockers, we need to review the basic physiology of the neuromuscular junction. The neuromuscular junction is the interface where a motor neuron meets a skeletal muscle cell, otherwise known as a myocyte. This region is comprised of three key parts. First, we have the axon terminal of the motor neuron. On the cell membrane, you'll find voltage-gated sodium and calcium channels. Next, we have the motor end plate on the skeletal muscle cell. Last, we have the synaptic cleft, which is the space between the axon and the motor end plate. Just like in all other synapses, the presynaptic neuron releases neurotransmitters that interact with membrane-bound receptors in pre- and postsynaptic locations. In the case of the neuromuscular junction, the neurotransmitter is acetylcholine and the receptors are nicotinic. There's a prejunctional and a postjunctional nicotinic receptor, and we'll detail the importance of these shortly. For now, understand that the postjunctional nicotinic receptor is comprised of 5 protein subunits that align circumferentially around an ion-conducting pore. The open or closed state of this ion channel determines whether ions are allowed to pass through. Notice that there are 2 alpha subunits, and we'll reveal the importance of these shortly. Now that we understand the anatomy of the neuromuscular junction, we can examine how the neuron communicates with the myocyte. At rest, the neuron maintains a polarized state called resting membrane potential. Said another way, the inside of the cell is relatively negative and the outside of the cell is relatively positive. An action potential, generated by the central nervous system or a peripheral nerve stimulator, depolarizes the axon terminal. This activates the sodium channels, allowing sodium to enter the neuron. The influx of positively charged ions reverses the distribution of electrical charge so that the inside of the neuron becomes relatively positive with respect to the outside. So, why is this important? Depolarization of the axon terminal turns on voltage-gated calcium channels in the presynaptic neuron, allowing calcium to diffuse into the nerve. Once inside, calcium initiates the mechanism that triggers vesicle mobilization and subsequent release of acetylcholine into the synaptic cleft. Once inside the synaptic cleft, acetylcholine diffuses along its concentration gradient towards the postjunctional nicotinic receptor. When 2 acetylcholine molecules bind to the 2 alpha subunits of the nicotinic receptor, the receptor's ion-conducting pore opens, allowing for the influx of sodium and calcium and the efflux of potassium. Accumulation of positively charged ions inside the myocyte generates another action potential that ultimately depolarizes the rest of the skeletal muscle cell, culminating in muscular contraction. It's important to note that some of the acetylcholine in the synaptic cleft stimulates prejunctional nicotinic receptors. This process increases the mobilization of acetylcholine stores so that the neuron is ready to be stimulated again. This physiology will become important when we discuss fade during train of force stimulation. For every ligand-receptor interaction, we need an off switch. We've already stimulated the muscle to contract, so now we need a mechanism to make it relax. Acetylcholinesterase is strategically positioned around the pre- and postsynaptic nicotinic receptors, and its job is to hydrolyze acetylcholine as soon as it unbinds from these receptors. Acetylcholine is metabolized to choline and acetate. Choline is pumped into the presynaptic nerve terminal where it's recycled and used to synthesize more acetylcholine, while acetate just diffuses away from the neuromuscular junction. The mechanism we just described ensures that the process of muscular contraction can be turned on and off like a light switch. But what do you suppose would happen if acetylcholine stayed bound to the receptor for a longer period of time? For those of you thinking ahead, this is exactly how depolarizing neuromuscular blockers such as succinylcholine produce muscular paralysis. Succinylcholine binds to the postjunctional nicotinic receptor and depolarizes the motor end plate. But unlike acetylcholine, succinylcholine remains bound to the receptor for a much longer period of time. This explains why you see fasciculations followed by flaccid paralysis. Muscle tone returns after a sufficient quantity of succinylcholine has dissociated away from the receptor at the motor end plate. What about non-depolarizing neuromuscular blockers? These agents occupy the nicotinic receptor and prevent acetylcholine from depolarizing the myocyte. This process is called competitive antagonism.